Hi everyone. Today we're talking about viruses, and I think really timely given what's going on in the world right now. Viruses are a really special group because actually they're not considered to be living things, but we do sort of consider them in terms of species and different things like that. And we know, given what's been going on in the world right now, viruses have a huge effect on living things. We obviously see that now with COVID-19, but we've always had viruses, they think, since the beginning of time. So we're going to discuss a little bit about that today. The really obvious reason to study them is because they are disease-causing agents, and another term for that is pathogens. Bacteria can be pathogens as well. So can other things like fungi can be pathogens and so forth. But viruses are really important as a pathogen because, as we can see, they can cause really widespread illnesses to happen. So I've mentioned over here these terms of epidemic and pandemic. Epidemic being localized to one area, pandemic being over a large area or even globally, which is what we're talking about with the COVID-19. So this diagram I've pulled over here is actually showing the influenza virus from last year, just to show you, and that's the common flu virus, just to show you a little bit about different strains. Okay, so you might have heard that term used before. A strain is basically a variation or a type of a virus. If, for example, you had a vaccine last year, that vaccine might be outdated because actually the virus continues to evolve, which is one of the reasons we have to get a flu shot each and every year. So we're going to discuss a little bit more about vaccines later. I want to just highlight for you really quickly the characteristics of living things. So these six things are common to everything that we consider to be alive. Okay. Now, the reason that I'm bringing this up specifically in the virus lesson is because we're going to see just here that actually viruses don't follow some of these characteristics. One of the things I'm asking you to discuss over D2L, and I've created a discussion post there for you to weigh in on your option, is whether we should consider viruses living or not living. So I'm going to walk you through a couple of things here, which is why we don't typically think that they are living. That's the most sort of accepted logic from the scientific community, even though you'll see like on a Lysol brand or something like that, like kills 95% of viruses or whatever the case might be. Viruses aren't actually alive, so you're really just destroying them. You're not actually killing them. So just something to think about. So a couple of facts here. So viruses are very, very simple. They're not made of cells. Instead, all they have basically is genetic material, which I mentioned here can be DNA, which is what we have, or RNA, which is a single-stranded version, for instructions. So they have their genetic material covered by a protein coat, which we're going to see there's some variations of how that protein coat can look in just a few minutes. But the key here is that they don't have cytoplasm. And so without cytoplasm and without a cell membrane, you are not considered to be a cell. So immediately just from that designation, this first characteristic is not met by viruses. So something to consider. They are very, very tiny. So when we're thinking of how small a human cell is, thousands upon thousands of individual viruses can fit in just one human cell. So they're very, very small because they don't have any cytoplasm. They also cannot grow or reproduce if not infected in a host cell, which I'm going to show you in just a minute. They also don't have a metabolism. And what that means in like really general terms is they don't produce energy, they don't eat, and they don't make energy from that food. And they also don't make any waste. So from that designation, we would say they have no metabolism of their own. As I mentioned with reproduction, they essentially rely on a cell that they've infected in order to get the things that they need. So we'll talk more about that after. Um, and this statement I've put here, viruses are always infectious. Okay. What that means is in order for them to actually propagate and increase their numbers, they need to be inside of a cell. So on a surface like a doorknob and so forth, the virus is essentially just sitting there dormant. It's not able to actually reproduce itself. But the moment that it gets into your body and then infects your cells, it will start to reproduce and create more and more of itself, essentially. So more about that on the next couple of pages. Now, I just want to mention that they are sort of important in a way because out in the ecosystems, they actually control population growth. I'm not talking so much about human populations, although that is true as well, but they also can infect things like other animals, for one, as well as plants. So that's helping to actually control population size. The last little point I put here is that viruses are actually very important for gene therapy. I'm not going to talk too much about that today, but we study that in Unit 2 when we look at genetics. So we actually use viruses as a tool to help treat people and create sort of individualized medication. So we will talk about this in the second unit. We'll just leave it at that for now, though. So next up, we'll just talk a little bit of the origin of viruses. So as I mentioned, viruses are not cells. And so there's a lot of sort of theory and there's a lot of inferences and hypotheses that we use to actually sort of figure out where they came from. And what's really interesting about viruses is because they're not cells, they actually could have just come together sort of by chance. So their DNA, RNA could sort of just come together in a chance sort of way and create sort of these viruses that we now see today. They also have this theory that perhaps they were a cell at one time, but over time that trait evolved away. They think that viruses are far older than living things, cells, in other words. And they also think that perhaps viruses were something of a precursor to cells. So they think that perhaps these viruses became cells over millions and millions of years. So all of these that I'm mentioning here, they are theories. They are what people think may be the origin or how viruses came to be. So that's just some information about that. A bit of classification stuff. I'm not going to read through all of these. I'll leave that for you guys to do. But just to mention that we still do classify viruses. So I mentioned with the bacteria lesson that it's sometimes useful to group together bacteria that perhaps cause a similar type of infection. That way we know how to best treat them. And the same thing is true here, although we don't try to kill viruses like I mentioned before, but even for design of things like vaccines, knowing a little bit more about the virus is very helpful for that. We also have things called antiviral medications, which try to stop the virus from actually infecting the cells and reproducing within them. So definitely important to know that. So I just want to mention a couple things about the structure. I mentioned earlier, viruses really simple. They've got the capsid, which is that protein outside, and the genetic material, which could be DNA or RNA. So I'll just show you really briefly here. These diagrams is just examples of different types of viruses. So some you might find familiar, like HIV. We've heard of that virus before. Bacteriophage, these are viruses that infect bacterial cells. And the reason for this blue arrow being here, you can see where the viral DNA is up top. Basically, this tail part of the virus will almost like sit on the cell and it will inject that viral DNA, and that's how it infects a bacterial cell. So each and every one of these are slightly different. I mentioned just above, some have an envelope. And the envelope is essentially something they pick up from the host cell. So you can see with the HIV virus, it has sort of an additional part on the outside. In addition to its capsid, it also has the envelope. Now, one thing that's really dangerous about this, and I'll kind of show it on this diagram, is this virus actually took some of the host cells outside, right? Took some of the cell membrane from there. So I mentioned here, but this becomes very difficult for your immune system to fight off because what your immune system is constantly do is like scrolling through, just finding anything that doesn't belong. But unfortunately, with this envelope, the immune system might see this and actually think it's totally normal. It thinks it belongs to your body. So some of these viruses, HIV being one example, they can lay dormant. Your immune system just doesn't react to them because it doesn't realize yet that there's some type of infection. So again, there's lots more than these examples. I just use this picture because it shows nice diagrams of each one. So I'll let you explore the ones that interest you further. The next part we'll talk about is actually their infectious cycle. So we don't call this a life cycle because I mentioned before, we don't consider them living. So as such, we just say it's a life cycle, infectious cycle. And so, as I mentioned, it's only activated when they infect a cell. And I mentioned that term host. So the host just means whatever organism they are infecting. So we have two sides to this cycle. I'm going to talk through the diagram, but you're going to see right underneath this, there's almost like steps involved. And because it's cycles, you can start from any one point. I'm going to actually start from right here in my explanation. You can see a virus has attached to the surface of this cell. This is an example of a bacteriophage. That's the shape that we see. And we can see that this little red piece of DNA has been inserted. And watch what happens over time is that that little red piece, the viral DNA, got inserted into the host cell's DNA. Now, the issue with this integration step, is that the cell is just copying its DNA as normal. You'll remember from grade 10, during the S phase of interphase, the cell just copies its DNA and starts to divide, right? This is a very similar process. The problem being what I mentioned before, the virus has essentially integrated into the cell and it's sort of tricking the cell, so to speak, into copying its viral DNA because the virus doesn't have the capability to do that on its own. So once this is inserted into the host cell, it just continues to copy. And so when we get down here to number 5, you're going to see the cell is dividing, creating new cells, but now it also contains viral DNA. And we spoke about bacteria in the last lesson. They are able to reproduce by binary fission, which is great. Means they can make lots of exact copies of themselves. But the problem is, if there's a viral DNA in there, they're also not going to remove that. They're just going to continue on copying the same thing over and over and over again. So over here, number 6, it's basically many, many cells that would have been produced with this. It's not just the one cell, but of course, we're not going to draw millions of cells for the diagram. Now, what happens in number 7, 8, 9 here, number 7 and 8, essentially, the virus has now tricked this cell into making more viruses. We know that DNA is the instruction manual, and so this viral DNA is actually telling the cell, okay, let's make lots of viruses. So in number 7 and 8, it's essentially assembling these viruses. Now, what happens in step 9 is I mentioned before, thousands of these viruses can fit in one cell. But just like a balloon that got overfilled with water, if this becomes overfilled with viruses, it will actually burst the cell. And we call that bursting lysis. So lysis being cell bursts. Now, I mentioned it right here. The cell will die from this, okay? The cell cannot repair this bursting. So what ends up happening is that you actually see damage to the cells. And the example I'm going to give you of that is the herpes simplex virus. That is the virus that causes cold sores. So some of you might have experienced this before, is that suddenly you're going to have an outbreak. So essentially the cells start bursting open and that's what we see as a sore. And they're often hard to get rid of because unfortunately, as these viruses are bursting out, they're also infecting nearby cells and the cycles continue. So it's often hard to treat those kinds of things because as I mentioned before, this virus is essentially tricking the body into, moving on elsewhere. So the immune system can't fight them as efficiently. So I'll just skip over this part because this is what I've just described. You can read it again. And this is where I was saying about the herpes simplex virus. So when it's in that lytic cycle, where that lysis occurs, it's going to destroy cells. And I'll just mention basic backtracking. This is usually activated by a stressor. So, oftentimes when people get cold sores, it could be an environmental stressor such as what we consider stress. Sometimes it's when the person gets sick otherwise with some other type of cold or flu or so forth, and so on. So, when something in the environment of the cells changes, it sort of triggers this entry into the lytic cycle, which is where we start to see the actual sores. So that's kind of one common example of that, and it applies to other viruses as well. Now, last thing I'll just talk about here is transmission. So I think one thing the COVID-19 situation has taught all of us is how important it is to wash our hands and wear our masks and so forth. And the reason for that is that many viruses, especially the ones that infect humans, they travel through a vector that is either airborne or by droplets. And so I mentioned before, viruses are really simple. They can't just get up and move. They don't have the machinery in order to do that. So we have to be really careful to sort of avoid these types of things. Insects, of course, coronavirus that we know does not travel that way, but you do have other types of viruses that travel within insects, especially that when mosquitoes bite you, they transfer it from point A to point B. So those are types of vectors, and as I mentioned before, other diseases can also have vectors. It's not just based on viruses, but that's just a common one that we see. So I want to just briefly talk about the immune system and vaccines. We know we have vaccines for lots of different viruses existing. It's very difficult to make a vaccine, so I am oversimplifying it by a lot. But I want to just give this as a really quick intro into how our body defends itself. So our body makes what are called antibodies. And antibodies are small molecules that protect against antigens. So I'm going to refer to this red and green picture. Here is a pathogen. I mentioned earlier it could be a virus, it could also be something else. An example for me is a pathogen might be an allergen. Okay, so something that triggers my immune system to start fighting it. And so what happens is these little green antibodies, they stick around in your body and they actually will bind on. So you'll notice there's a very specific, almost puzzle piece fit. So this specificity is to say that antibodies are supposed to bind onto there and basically disable the pathogen. The other thing that it does, which is really cool, is that it's going to attract other cells from the immune system to come over and actually destroy these. So the main cell doing that is called a macrophage. And macrophage will actually gobble this up whole. They're very big cells and will basically digest it within itself. And that's what actually causes, if you've ever had a sore on your skin, that's what actually creates pus. So those macrophages, once they've destroyed all this bacteria or viruses or whatever the pathogen might be, they themselves are also destroyed. And what we see that come up as on the surface is pus. Okay, so that brings us to vaccines. And the goal of vaccines is that before you get infected, you can almost like prime your immune system. You can get it ready to respond. And so, what the vaccine usually does is put a little piece of the virus, a small piece of those surface antigens, into your body, allowing your immune system to create antibodies for it. Other times, it can be a weakened form of the virus, which they'll put inside, and the same thing will happen. Your body will see it and try to respond to it. So, a good example of this is you might have heard of now there's a chickenpox vaccine. So, that vaccine, they put basically a little bit of the chickenpox into the person and they allow your body to create antibodies for it. And then with chickenpox, hopefully, you shouldn't get it again because you already have the antibodies. And that particular virus does not change too much year after year. So that's why I said here some vaccines are permanent, others are not. right? I use the example of mumps and measles, which many of us get when we're first, as infants, when we're first born. They don't change too much year after year because there's not much transmission of them anymore. But something like the flu virus, it can evolve quickly because it's constantly moving from person to person and globally as well, which is why we need an annual vaccination. And so, what happens annually is that scientists try to predict what the flu surface will be, what the antigens will look like. They try to predict that a year in advance so they can create a vaccine and hopefully we can get that vaccine before actually getting sick. So there are some dangers associated with this. I've posted a video link to show you just a little sort of summary of vaccines. But if I'm putting in a little piece of a virus into someone, they might actually start feeling a bit sick. So I've experienced that myself as well, where I get a flu shot and then for the next couple of days I might feel almost feverish or feel almost like I have a bit of the flu. But that's just because my immune system is working to respond to those antigens. So the last thing I'll just mention here is applications of viruses. Now, I mentioned it before. We're not going to go into information and details about this right now. We're going to actually save that for the genetics unit. But the reason I've put it here is just to say that all viruses are not necessarily a bad thing. We have learned to manipulate them. We've learned a lot from experimenting with them as well. But they're a huge, huge tool that we're using for gene therapy, which I was mentioning earlier. That's that personalized medicine. So just something to consider that it's not all bad. We do actually use them as tools for biotechnology.